So the role of your DNA.
Remember our procrastination party? If you said it.
Okay, so the role of your DNA is to code for proteins, and this is covered by a process called the central dogma of biology that we'll be getting into probably around February or so. Central. And you've heard of this before. We're just going to get into more detail, but this is the idea that your DNA is used to code for messenger RNA, and then messenger RNA is used to code for proteins. And we'll get into all that. But basically, I just wanted to reinforce the idea of what your DNA is actually used for. Now, you have roughly 6 feet of DNA inside of every single nucleus, inside of every single cell. That's a ton. So if you imagine stretching your DNA out 6 feet's about my wingspan, if that has to be coiled up and packaged into an individual microscopic nucleus, which seems like quite the task, but your cells are up to it. So basically what this flowchart here shows is the formation of a chromosome, and basically as you move to the left from this final structure that you probably recognize as being a chromosome, we are zooming in further and further and further into its structure. So at its base, a chromosome is just coiled up DNA. So I'm just going to go down and we're just going to label these things. This would be DNA. Okay. Now, you'll also see here the size, so like the scale that we're working off of, so like 2 nanometers, that's extremely small. So DNA is at the scale of about 2 nanometers. Now, if you take DNA, the first step in forming a chromosome is it's going to be coiled around proteins called histones, and that's going to form a structure called a nucleosome. A nucleosome. Okay? And nucleosomes are made up of DNA and histone proteins, and those together form a nucleosome. The histones, think of it being like a spool, and DNA being like thread. And if you imagine like winding up thread around a spool, that's essentially what we see happening here. Now these proteins, they are actual like little enzyme proteins. They're going to be actively coiling DNA around them. It's an active process that requires ATP. It's not like just something that magically happens. Okay? If you take a whole bunch of nucleosomes and coil those together, then you end up with a structure you've heard of before, chromatin. Chromatin is DNA that's been loosely wound around these histones. They're not condensed very tightly, and most of the time your DNA is in the form of chromatin because this is the form that your DNA is in during interphase. So during interphase, your DNA is in the form of chromatin, and it can be used to code for proteins at this stage. The more it condenses, though, the less available it becomes. Okay, if the chromatin continues to condense, then we will see the formation of chromatin loops. And then it will condense even more, at which point you call it condensed chromatin. I know, right? And then finally, if the condensed chromatin wraps up into its final structure, you end up with the familiar chromosome. So, a chromosome is just tightly coiled and packaged DNA. That's all it is. And it's wound up with those histone proteins. That's the mechanism that keeps it tightly wound up and packaged. You're used to seeing DNA in the form of a chromosome, but normally it's going to be in the form of chromatin, loosely wrapped around these histone proteins. Okay. The next thing I want to do is we're just going to list out like some general facts about chromosomes. So this is our list of general chromosome facts. Here we go. General chromosome fact number 1. They're made up of DNA wrapped around histone proteins. Okay. We do need this space right above these, so maybe like section this off so you don't spill over into that area. So made up of DNA wrapped around histone proteins. Okay. General chromosome fact number 2 is chromosomes allow the condensing. I don't know how to spell condensing, don't judge me. Is that right? It just looks weird. 6 feet of DNA into a microscopic nucleus. So again, we're able to wind up that 6 feet of DNA into a nucleus, which is microscopic, which is pretty impressive. Now, chromosomes are not always present. Super common misconception. So, chromosomes, when do they form?
After.
Chromosomes form during?
The end of interphase, right?
Yeah, it's one of the first things that happens during prophase. So you can just say during prophase. So chromosomes form during prophase of mitosis. Before that, so during interphase, DNA is in the form of chromatin. Now, the reason this matters is because chromosomes are packaged up so tight that the DNA is not going to be accessible for coding for proteins when it's in the form of a chromosome. So right below that, I'm just going to put a little asterisk here and say when in the form of a chromosome. DNA cannot be used to code for proteins. Okay, so think about what that means. So after prophase, can any more proteins be made within that cell? No, they can't. So that means during interphase, the cell has to prepare. Sorry, I'm trying to, it kind of spreads out over a big of space. The cell has to prepare for the actual division of that cell by producing all of the things that the cell is going to need to divide. I like to think of this as like if you were moving, so you were going to be moving into a new house, and you like packed your cell phone or something else that you really wanted, and then the cell phone ends up in a box gets taped shut, puts on the moving truck, and now that phone is packaged up in a chromosome. It is no longer accessible to you. You can't get to it even if you wanted to, and you're not going to get your phone back until you unload or unpack the chromosome box in the form of chromatin at your new house, right? So in the new cell. So once that chromosome forms, the DNA is not available to code for proteins any longer. And it won't be until you create the new cell. So again, these cells have to front load everything. All the stuff that needs to be made in order for the cell to divide has to be made up front during interphase. Okay, the last thing I want to put on our list of general chromosome facts is that generally, depending on the organism, but usually there's going to be two types of chromosomes. There's going to be the autosomes and the allosomes. Autosomes determine what organism it is. For instance, chromosomes 1 through 22 make us human, right? We have a set of allosomes, though, chromosome 23, X and Y. These determine the sex of that organism. Typically. Again, this is for sexually reproducing organisms like humans. So what we see is a set of autosomes that every organism will have within that species, and then a set of allosomes that only particular sexes of those species will have. So like with humans, you all have chromosomes 1 to 22. Chromosome 23, you'll either have two X's or an X and a Y, and that'll determine the sex of that. So your autosomes make you human, your allosomes make you a male or female human. Okay. Let's see. We're probably gonna have to cut it off there because we only have a couple minutes left. So tomorrow we're gonna pick up on the idea of homologous chromosomes and sister chromatids.